Today we're going to talk about the cell membrane, why it's important, its role, and what makes up the cell membrane. It's frozen. Okay, so the cell membrane filters or regulates really what goes into and out of the cell, which seems very simple, but it's actually a very, very important job, and there's a lot that goes into that job. It's semi-permeable, which means it allows for the uptake of key nutrients and the elimination of waste, but it also maintains a protected environment by not allowing everything to go into or out of the cell. So semi-permeability means it lets some things in or out, but not everything. And this allows for compartmentalization. So organelles, for example, have membranes, and we know that every organelle has a specific function. The Fluid Mosaic model is the current model of the structure of the cell membrane. So there's two important words, and chances are you will be asked about these words on a test or even an exam. So the first word is fluid, the second one is mosaic, and we're going to talk about what those both mean. So specifically, membranes are not rigid. So we typically draw them as like a circle, and we would consider that like a solid circle, but they're not. They move and they have like a fluid motion. So they contain lipid molecules, which we've talked about, and they also contain proteins. So a lot of times when we talk about the lipid bilayer, we're literally only talking about the lipids, but there's actually a lot of proteins and other structures embedded in that membrane, and we're going to look at that. And in that membrane, the proteins are actually able to move freely within that membrane. So the fluid part means that lipids and proteins are free to move laterally within the two layers. So let's look at a picture. So this is a classic diagram. This one's out of your textbook, but this you'll find as a perfect representation of the perfect membrane with all of the things in one place. So we have here our lipid bilayer, and those are the phospholipids with their polar heads and their nonpolar tails. So the polar heads are going to be hydrophilic, and so we're going to see them pointing outside the cell. So this is outside the cell where it's going to be an aqueous layer, or in the cytosol, which is again an aqueous layer. And then the hydrophobic tails naturally point inward because they're hydrophobic. They want to be away from water. So remember, this happens naturally. And then we have all of these proteins embedded within the membrane. And we're going to talk about the different ways that they're embedded. There's different names, and they typically have different functions. But let's just take a peek here. And I want you to become familiar with this diagram. And tomorrow you will be labeling a diagram like this, but I really want you to spend some time drawing your own representation. So on a test, you may be asked to draw a lipid bilayer with certain types of proteins within them. So let's talk about them, and then we'll go into more detail throughout the lesson. So we have these proteins that span the membrane. So they're going like from one end, the outside of the cell, to the inside of the cell. These are integral membrane proteins. And that means that they're going like literally like through the membrane. So there's some parts that are out and some parts that are in and some parts that are going through the actual membrane. And you can think that there's going to be amino acids out here that are hydrophilic, and same here. And then there's going to be amino acids in here that are hydrophobic. So it actually helps with the folding of the protein and the structure and the function of that protein. So this protein would naturally want to be in this place because we have amino acids that want to get away from the aqueous environments. Okay, and then we have peripheral proteins. These ones are attached to the inside or the outside. Usually it's the inside of the membrane, but they're not going through the membrane. So they're completely hydrophilic. And again, they have a specific function. And then we have things that, like carbohydrates, for example, they're attached to membrane proteins. And so these have a specific name. And we have other things like this is called a glycolipid. So we have a lipid attached to, so we have the carbohydrate attached to the lipid bilayer. And we have cholesterol here, which remember cholesterol is amphipathic, so it behaves like a lipid. And so we're going to have the polar end hydroxyl group of the cholesterol and then the hydrophobic rest of the structure. Okay, so those are like, this is a overview, and we're going to go and talk about how they have specific functions. Okay, so the lipids, we've talked about this. This is review. They exist in a double layer, the bilayer. They're actually less than 10 nanometers thick, which is extremely small. Millions of times a second, lipids are moving in one way or another. So they are either just like vibrating, or they'll actually flip back and forth. So we see here, they will actually just kind of exchange places with each other. They can also move sideways, flipping with each other. They can also spin around with each other. So there's a lot of movement, and again, that's the fluid part. Okay, the mosaic means, for those in art class, mosaics are usually consisting of a lot of different parts of things. So that's where the mosaic part of the name comes from. So we have a wide assortment of proteins that exist in the cell membrane. And they have different functions. So we have transport into or out of the cell of nutrients or waste products, attachment of things, so attachment of the cytoskeleton inside or attachment to other cells on the outside. And we even have, again, enzymes for biochemical pathways that will be found within the lipid bilayer. They are larger than lipids, and they move more slowly within the lipid bilayer. Those that anchor the cytoskeleton won't actually move, and we'll talk more about that in a bit. So again, I just want to show this diagram because I just love that we can see the actual like opening of that lipid bilayer, and we can see we have proteins embedded just in the top layer, just the bottom layer going through. So again, there's a lot of things that make up the cell membrane. So there's two types of carbohydrates that we're going to talk about with the lipid bilayer, and the first one is glycolipids. So this is where we have any membrane lipid, so in this case right here, that has a carbohydrate attached to it. So the carbohydrate chains are shown as like, literally it looks like little sugar molecules attached and branching. So that's typically how you will see a carbohydrate represented. And so we have the glyco part, as in we can think of glycogen as a chain of carbohydrates, or a chain of sugars. So we have our glycogen, glyco, and the lipid, because it's attached to a lipid. The other one is a glycoprotein. Same sort of thing, where we have a protein embedded in the membrane, and it is bound to, again, a carbohydrate. So we have the glyco part, as in glycogen, as the representation of our carbohydrate, glyco, and then the protein. Both tend to play a role in cell recognition and cell-to-cell interaction, because this is typically outside of the cell. And so the cell recognition is where it might interact with another cell or a signal from another cell. And same thing, there might be some sort of communication between cells. And so these will either tell another cell what type of cell this is, or it might be giving some sort of information to another cell. And that's something you would learn more about in further years of university. So in cell recognition, we have the proteins in the plasma membrane. And like I said, they're acting almost like a fingerprint, telling other cells outside of that cell what kind of cell it is or what its purpose is. So it's almost like a fingerprint, which is really cool. So other cells might have direct contact with other cells, and they do this through gap junctions. And anybody who had me as a grade 10 teacher knows I love gap junctions. So random. They're also called the plasmodesmata in plants. I won't ask you about that. There's chemical communication via signal molecule. So we may have hormones or other molecules sent by other cells that will be communicating with different parts of the body. And we have, this is really cool, looking here. So this is the lipid bilayer of one cell, and this is the lipid bilayer of another cell. And we have these channels, so they're called gap junctions, that line up with the other cells, and they can actually allow movement of molecules, either like signal molecules or even nutrients, into other cells. So the cells they're not an island. They are connected to each other, and it is through these gap junctions, and these are all made of proteins, so these channels here. So what's really interesting is neuroscience, the whole field of it. And so we do come back to this in Unit 4 when we talk about the brain and its structure. But it's really cool to look at just this part that is involved in this part of the course. So the myelin sheath is part of the brain. And myelin is a substance that acts to insulate nerve fibers. So here we have a nerve, and remember we kind of looked at, we might have looked at this in grade 10 or 11, where we have the neuron here, and it's got all those branching to be able to communicate with so many other cells. And then it has this long axon here, which will, again, make connections with other cells. So here it's receiving signals, and then it's sending signals through this axon. And this gets to be protected. And we'll talk about the importance of that protection and how it actually really helps the neuron send really fast signals. And this protection here is the myelin sheath. And it's actually made up of 82% of lipid, which is a really high content of fat. And what's neat is it's a modified and extended plasma membrane. So we see here, this is a cell, and we have this really long extension of the plasma membrane. So it's just like a really high amount of lipid bilayer, and it wraps around the axon, and this allows electrical impulses to transmit quickly and efficiently. And we'll talk about how that works in Unit 4. Multiple sclerosis is one of the best known diseases where demyelination occurs. So those patients will have missing a lot of the myelin sheath, and therefore can't transmit the signals, and therefore their motor skills decline rapidly. Okay, membrane asymmetry. So asymmetry means it's not the same on both sides. So proteins and other components of one half of the lipid bilayer differ from those that make up the other half of the bilayer. So looking here, we have, for example, proteins on this end and here, but we only have a little bit of some protein or a transporter on this end. So they're not the same on both sides. This reflects the difference in functions performed by each half. So on this half, for example, it might be signaling or communicating with another cell or transporting something in. This side, for example, might have something to do with, and we're going to kind of see an example later on in the course where something like this happens, where we're going to have some sort of enzymatic reaction happening, or, as mentioned earlier, this holding the cytoskeleton together. Okay, so we're going to talk about the interface, so that's the inside of the membrane. Typically, we have cytoskeleton binding to proteins on the interface, so that's a big function of these proteins, those peripheral proteins. They are holding onto that cytoskeleton to keep everything in that scaffolding in place. The outer face, we have our excuse me, our glycolipids here, and we have our glycoproteins here. So glycolipids and carbohydrates attach to proteins on the outer face for signaling. So remember, communicating with other cells. We have hormones and growth factors that bind to the receptor proteins that are found only on the external face or the external surface. This may trigger changes to different protein components on the inner surface. So binding on the outside might create a change on the inside. And then this would lead to subsequent reactions within the cell. Hey, interestingly, again, leading up into Unit 4, we'll talk more about this, but I just like to tie it all together. So we have serotonin, which is a hormone and more commonly known as a neurotransmitter. So neurotransmitters are communication between nerve cells and the rest of the body. This hormone is used for stabilizing mood and feeling of well-being and happiness. So serotonin is a fantastic hormone. We want more of it. Lack of serotonin is often associated with depression, anxiety, and insomnia. So medication that treats depression and anxiety will often help regulate serotonin levels. And we see here we have a neuron which is releasing serotonin, and this is its target cell. And we'll talk again more about that in Unit 4. So serotonin is made from tryptophan. Remember what tryptophan is? I'll ask you tomorrow. Mostly, interestingly, serotonin is produced in the digestive system. So again, there's links to how healthy our digestive system is and how happy our mood is. It is involved in sleeping, eating, and digestion. So if you need a boost of serotonin before we, if we just feel we need a small dose of serotonin, I'm not saying replacing medication, but often our diet has a lot to do with how we're feeling. And so increasing our leafy greens, sunflower seeds, and there's a whole list of them there. White meat, such as turkey, also contain the amino acid. The body can have a difficult time converting it to serotonin, however. So bright, natural light, exercise, and meditating are known to give a healthy boost of serotonin. So phospholipids, again, we've gone over the structure, so we're not going to go too much into it, but we have two fatty acid tails linked to a glycerol, shown here. And on that third carbon of the glycerol, we have a phosphate group and specifically some sort of polar head. So in this case, we have choline. So this is phosphatidylcholine. So the name of the phospholipid will depend on what polar molecule is right here. So in this case, choline, phosphatidylcholine. You don't have to know how to name these. That's definitely further in university. And remember, the head group is charged, it is polar, and the tails are hydrophobic. And so again, this would be an amphipathic molecule. And then again, because of this structure, we have this spontaneous creation of a bilayer when placed in an aqueous solution. Okay, so the fluidity of the membrane is influenced by the composition of the lipid molecules that make up the membrane, and it's also the temperature. It has an effect on the fluidity. So fatty acids with the saturated hydrocarbons are going to be straight in shape and they'll pack more tightly. So we talked about this earlier in the unit. Unsaturated fatty acids have double bonds and they have kinks in them, so they'll pack more loosely. And we see here, these are packed nicely, and for example here, and so they're going to be nice and stacked up. These ones, It looks like a disaster. So these ones have lots of kinks in them, and they're going to be more fluid. So these would be fluid at room temperature. So we're looking at our plant oils, so olive oil, vegetable oil, whereas these are going to be more viscous. So they're going to be more solid at room temperature, so like our butter, for example. So membranes remain fluid over a large range of temperatures, but at lower temperatures, phospholipids in the bilayer don't have as much kinetic energy. And so they're going to cluster together more closely, and they're going to increase their intermolecular interactions. So this will decrease membrane fluidity. If low enough, the lipid molecules become closely packed, and the membrane forms a highly viscous semi-solid gel. So again, thinking about butter at room temperature. The more unsaturated, so the more double bonds, the lower its temperature can be. So butter is highly saturated versus olive oil, which is highly unsaturated. So this would be our butter, and this would be our olive oil. Sterols, like cholesterol, for example, are membrane stabilizers. So they restrain the movement of lipid molecules. So they're going to reduce fluidity. So at low temperatures, they are going to occupy the spaces between lipids and prevent them from moving. So cholesterol is, as we learned, a sterol found in animal cell membranes. So they play an important role. So they're important for us. We just can't be consuming too much of them. So our body will produce the amount of cholesterol that we need. We don't need to consume it. And it is going to create protection for the cell by, again, occupying this space. It reduces permeability. So it prevents things from coming in or out. So reviewing, again, and again, we're just going to overview this, the role of transport proteins in the cell membrane. One is transporters, and we know I spend a lot of time studying them, so I try to not go too much into them, but we have 2 lessons. It's curriculum, not me. So the first one are protein channels. So this allows compounds to pass through. So this would be just a channel that allows molecules to go in or out. We also have carriers that will actually change shape once they bind their solute or their substrate, and sometimes they require energy, sometimes they do not. We also have enzymes that can be embedded in the membranes, and this is really cool. So this is within our gut, so in the lining of our intestines. So they're called brush border cells. So these are on the villi of our small intestine. And we'll have an enzyme actually just embedded in that cell membrane. And as we have nutrients going past, for example, maltose, the enzyme will bind maltose and break it up into the 2-glucose, and those can be transported by a different membrane protein transporter, and the glucose can be transported in. So it's easier to transport in the glucose and not the maltose. So it has to be broken down by an enzyme embedded in the membrane and then transported by a different membrane protein. It's cool. Okay, and then we see similar things in respiration and photosynthesis, which we learn about in the next unit. Okay, again, we also have signaling between cells. So we have signals, which could be chemicals or hormones that will bind the membrane proteins shown here, which will cause some sort of reaction down the line within the cell. And again, this would be something you learn about in the future. Okay, we also have attachment and recognition, so proteins exposed to both internal and external environment. So we have in the external environment, we have our cell-to-cell recognition, or binding to the extracellular matrix, so keeping all the cells, not just one cell, but all of the cells that might be part of an organ system in its place. And we have on the internal attachment points for the scaffolding of the cytoskeleton. So we have integral membrane proteins, and there's essentially two types. So we have our peripheral membrane proteins that are attached to one part of the lipid bilayer, but not spanning it. And then we have the transmembrane proteins, and these are spanning the entire membrane. So you're going to have parts on the outer part of the membrane going through the lipid bilayer, and then a part sticking out the inside of the membrane. Okay, so thinking about the structure of the transmembrane or even the peripheral membrane proteins, we have to think about why or how that happens, and because it's going to happen naturally. So we can think about the structure of those proteins, and we can imagine any part of the protein that is within that lipid bilayer. So here this would be a transmembrane, and very simple. This is a very simple model of a transmembrane protein where we have our hydrophobic amino acids on the inside, and then we have hydrophilic amino acids on the outsides. Okay, peripheral membrane proteins are positioned on the surface of a membrane. So we're looking at these ones here, for example. So they do not interact with the hydrophobic core of the membrane. They're held to the surface by non-covalent bonds. So we can think of hydrogen and ionic bonds. And they usually interact with exposed parts of integral proteins. So this would be an integral protein, and this is exposed. So this would be a peripheral protein. Or directly with a membrane lipid, so shown here. They're mostly on the cytosol side, so the inside of the cell, and they are often part of the cytoskeleton, or again, binding to the cytoskeleton to keep that scaffold structure in place. So again, interacting with microtubules, microfilaments, intermediate filaments, so those are all just words from earlier in the unit. And they are proteins that link the cytoskeleton together. They hold some integral membrane proteins in place, so they're typically all structural.